Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. So Michelle, it's so good to see you again. I think we met 2 years ago when we had this Cancer Neuroscience Symposium in 2024. And so great to see you again. We're very excited to sort of build up towards the symposium in 2026 in February. And part of the goal here is to sort of get to know the speakers, so learn a little bit about you, the person, a little bit about your past and what were sort of the drivers that led to your research now, and then a little bit about your ongoing work, and then maybe some visionary sort of future-minded thoughts on where you see cancer neuroscience headed. If we could maybe start with, I think Kinsey had mentioned that you've done some pioneering work. I think it's well recognized in the field, the interactions between cancer and neurons, glial cells and neurons and gliomas. Maybe you could speak a little to how you sort of arrived at those early observations, and also from the perspective of being sort of a pioneer. What's your advice for young researchers who might also be, thinking about novel ideas, but maybe a little bit of hesitation in sort of pursuing those ideas? So you could sort of take us back to prior to that recognition that there's such integration in cancer neurons, and what you were thinking and what gave you the courage to sort of move forward.
I'm a neuroscientist by training. I studied developmental neurobiology and neuroplasticity mechanisms. And when I was in medical school, I was really struck by the spatial and temporal pattern of gliomas. These are cancers that happen in the developing nervous system at very specific ages in very specific anatomical places. And it seems so clear to me that these, really terrible gliomas of childhood, including diffuse intrinsic pontine glioma and other high-grade gliomas that happen in kids, adolescents, and young adults, were diseases of dysregulated neural development, and that we had to sort of to understand them and understand them well enough to develop effective therapies. We needed to approach them from that perspective. As a neurobiologist, it's really clear that the activity of the nervous system itself is one of the most powerful forces shaping development and plasticity. And so, from that perspective, I thought, well, gosh, what's known about the way that neurons influence gliomas? And really at the time, all that was known was a neuropathological observation from nearly 100 years ago by H.J. Shear that, glioma cells of many different types cluster in really tight microanatomical association around mature neurons. This is a phenomenon we call perineuronal satellotosis. And then they invade along axon tracts. And I thought, well, gosh, there's such a tight anatomical association. As a first principle of, neurobiology, the activity of neurons really shapes a lot of plasticity and development mechanisms. I've got to study this. I've got to see if activity influences the cancer. And so as a first step, as a postdoctoral fellow, I wanted to understand where DIPG and other diffuse midline gliomas arose from. What was the developmental process? What cell type were these tumors coming from? And discovered at that time that DIPG and many other kinds of gliomas actually most likely arise from precursor cells in the oligodendroglial lineage, in the myelin-forming glial cell lineage. And work from my lab and from many others over the last 15 years or so has really supported that idea, that that's the cellular origin of these cancers. And so when I started my lab, I thought, I'd really like to understand how nervous system activity influences these normal glial precursors. There was an idea at the time that maybe myelin was plastic and adaptable to experience, but it was very controversial and, not really clearly shown in vivo. And then at the same time, how do those really, potential mechanisms of plasticity, of glial plasticity, play into glial cancers. And so when I started my lab, I thought it seemed like a really simple set of experiments, that we would take tools from modern neuroscience, things like optogenetics, and use that approach to control neuronal activity in ask, how does that affect normal myelin forming glial cells during development and throughout adulthood and different circuits? And how does that in turn affect these glial cancers that we believe are arising from these precursor cells? So, started my lab now about 15 years ago. And the first experiments we did were, two different projects in parallel probing the effect of glutamatergic cortical projection neuronal activity, just as a place to start, on normal oligodendrocyte precursors, and how that we then discovered, shapes myelin, plasticity, promotes new oligodendrocyte generation, promotes the sort of changes in the myelinated infrastructure of a neural circuit that tune its dynamics and really strongly influence its function. And then how, the same kind of experimental setup, but in the context of patient-derived high-grade gliomas, and discovered that when a high-grade glioma is present, it behaves just as its normal cellular counterpart, OPCs, and in response to cortical projection neuronal activity, really increases its rate of proliferation, resulting in an overall increase in growth. And so since those first pair of experiments that we did, my laboratory has, continued for the last 15 years or so to study, neuron glial and neuron glioma interactions.
That's just so fascinating. You mentioned healthy versus cancer, and these interactions between the
Yeah.
neuronal and glial cells and neuronal and glioma cells. Are those differences both spatio-temporal as well as sort of secretome? I mean, how are you trying to define the sort of, you mentioned circuitry, how are you trying to define the interactions both in terms of soluble factors and also sort of the structural landscape of
Yeah.
this
Yeah.
interaction?
So one question is, does every neuron type influence healthy OPCs or their malignant counterparts in diffuse midline gliomas and other types of glial cancers like glioblastoma? And the toolbox in modern neuroscience, again, turning to techniques like in vivo optogenetics, allows us to control targeted populations really specifically, one subtype of neuron in one place in one defined circuit, and probe these different interactions. And so with respect to normal myelin plasticity, there actually is quite a bit of specificity. Only some neurons promote the proliferation of healthy oligodendrocyte precursor cells and myelin changes. Only some subtypes of types of neurons, so cortico-callosal cortical projection neurons promote myelin plasticity, but corticospinal cortical projection neurons do not, at least not in the healthy nervous system. Within, for example, the dopaminergic circuitry, there is a very strong effect of dopaminergic neuronal activity on healthy OPCs, but only in one part of that circuit and not in the rest of the circuit. And so there's a lot of specificity, and that makes sense in the healthy nervous system when we understand that these, relatively small changes, very finely tuned changes in myelin, really have big effects on the dynamics of a circuit. For example, coordinating, the arrival of two signals on a target cell. And that can be maladaptive when there are diseases of dysregulated activity or, too much activity or abnormal patterns of activity. These myelin changes can actually be strongly maladaptive and disease promoting. So the healthy OPC very, very finely regulates these activity regulated responses. The cancer tends not to invent anything new. It tends to hijack existing mechanisms of, physiology, but then to subvert them and use them for, the benefit of the cancer. So what we found is that in contrast to that, while a lot of the same mechanisms, there are such clear mechanistic parallels between normal neuron glial and malignant neuron glioma interactions are, holding true, the cancer seems to respond to just about any kind of neuron that we have studied through a variety of mechanisms. We see this in many different regions of the nervous system. There's some circuit specificity. Some neuron types have influences on glioma, at the business end of the neuron, if you will, at the end of the axon. Others have, effects through secreted factors that are, present both around the neuronal cell body and also along the axon. But every neuron type that we have studied so far has really robustly driven the proliferation, the growth, survival, invasion, all kinds of pathophysiological features of malignant gliomas.
Hmm. Interesting. I'm wondering, as you were speaking, I was thinking about the interactions, the very local interactions between the glioma and the neurons and the secreted factors. Could you speak a little bit more about some of the secreted factors? I know you've worked on BDNF and
Yeah,
neuroligin
yeah.
3 and
Absolutely, absolutely. And one important thing to remember is that secreted factors, neurons are very, very long. And so that the action of nervous system activity on a tumor is actually pretty brain wide because, distant neuronal cell bodies will project long axons,
that's what I
towards.
was wondering, the distal effects versus, versus the more central, very local effects.
Yeah. And so, I really think it depends upon number one, the mechanism, and number two, the neuron type. So, GABAergic interneurons don't have such long processes and they generally exert their effects in the local tumor microenvironment. Long range projection neurons can affect a glioma cell, from quite a distance from the cell body, but not a distance from the neuron. The neuron is, reaching out an axon, and either synapsing onto the glioma cell or secreting, other kinds of factors locally into the tumor microenvironment. So it's just important to remember the anatomy of neurons, means that, local means different things. So, but if we think about the cell body and we kind of think about the neuron in different sort of cellular compartments, there is, of course, the neuronal soma, the cell body. There's the sort of dendritic compartment of the neuron, and then there's the axonal compartment of the neuron. And some of the effects of neurons in an activity regulated way are through secreted growth factors, secreted paracrine factors. And we actually, that was the first mechanism that we discovered, and initially thinking, that must be a major way that neurons are influencing glioma cells. And indeed it is. If we, take a healthy brain slice and we collect the secreted factors when neurons are very active, moderately active, or silenced, then we can, discern that there are activity regulated secreted factors when placed on glioma cells that promote glioma cell proliferation, invasion, etc. We've gone into quite a lot of work to identify those factors. Of course, they're different. There's a different complement of growth factors from different neuron types, but starting with the, sort of glutamatergic neurons, we find that there are activity regulated neurotrophins like BDNF that, not surprisingly, strongly affect glioma cells. BDNF is one key growth factor that influences healthy oligodendrocyte precursor cells. And then there are some really unexpected things. So doing this kind of experiment where we collect conditioned medium from active brain slices or silenced brain slices and looking at differentially secreted factors, in a very unbiased way, when we do that kind of screen, we find, some unexpected, newly recognized growth factors. And one that we discovered now about 10 years ago is a shed form of a synaptic adhesion molecule called neuroligin 3 that, when shed and interacts with the glioma cell in a very powerful way that both promotes glioma cell proliferation, but also regulates gene expression of very interesting gene programs, and classes of genes, including a number of synapse-associated genes.
Do you think that there might be secreted factors that are not necessarily proteins, like lipids and maybe extracellular vesicle
Yeah.
containing factors?
Yeah, it's something that we're exploring right now. We had found in that initial set of experiments that we did, and we've repeated with multiple different kinds of neurons, the factors that are secreted, that if you take that conditioned medium and place it onto cultures of patient-derived glioma cells, those factors are heat sensitive, and RNase, DNase insensitive. So, we believe that they are proteins. Now, some of those proteins could be packaged in extracellular vesicles. That is a hypothesis and something that, we and others are exploring. There also could be really interesting other kinds of macromolecules. We know based on molecular size that the active factors, at least those that we can identify in that kind of conditioned medium experiment, are macromolecules, but there certainly are many different molecular types that may be acting more locally in a secreted way. We certainly know that there are a number of different neurotransmitters and neuropeptides that directly act on the glioma cell, both in paracrine and also in synaptic fashion. So, lots to explore. But this first class of molecules that we identified were proteins, based on their biochemical properties, and the two top ones that we were able to identify and validate are BDNF and neuroligin 3.
So these are a lot of direct interactions between the glioma cell and between the neuron, but we know that the tumor microenvironment also contains many immune cells. And I know that your lab is starting to look into that. And, working on microglia, my first thought is this is a cell type we know interacts with gliomas and we know interacts with neurons. So what do you see the role of the immune system, the vasculature, all of these other components in modulating their crosstalk?
Yeah, it's a great question. And actually, I didn't mention it, but activity regulated neuroligin 3 shedding is not mostly from neurons. It's actually mostly from normal oligodendrocyte precursor cells. So another glial cell type in the tumor microenvironment, which interestingly is a cell type that forms synapses with neurons. The OPC is always postsynaptic and expresses very high levels of this postsynaptic cell adhesion molecule, neuroligin 3. When we knocked out neuroligin 3 from different cell types that highly express it, we found that OPCs were the chief source of neuronal activity regulated neuroligin 3 shedding that then acts on the glioma cell. And so in that case, it's actually activity regulated, but not direct neuron glioma interaction. It's indirect through the OPC. So it's one great example of this point that you bring up, that there are clearly neuronal and neuronal activity regulated influences on myriad cell types in the tumor microenvironment, including microglia, astrocytes, oligodendrocyte precursors, mature oligodendrocytes, vascular cells, infiltrating other immune cells, and the crosstalk is complex. In the tumor microenvironment, particularly in the context of immunotherapy, I think this becomes extremely important. And we're currently focusing on understanding the activity regulated neuronal signaling molecule effects onto immune cells in the tumor immune microenvironment and how that influences anti-tumor immunity. So more to come on that, sort of an ongoing set of projects. But you're absolutely right. There's really complex crosstalk.
When we think more about this tumor microenvironment, you also think about whether or not the neurons are producing the same factors or different factors in the context of the cancer.
Yeah.
And because you were mentioning that cancers don't create anything new, they just hijack what's there. So is it just a matter of dosage of existing factors that are being secreted, or are there actually differences in the signature of secreted things?
Yeah. So I definitely think that the tumor alters, a lot of cellular biology in the tumor microenvironment. The factors that we've identified to date are normally secreted in an activity dependent way and have important roles in healthy myelin biology. But that doesn't mean that there isn't, new biology yet to be identified and discovered. And we have some ongoing projects that by 2026, perhaps I'll be able to, we'll be mature enough to talk about, but clearly that the tumor is influencing the response of cell types within the tumor microenvironment to activity in really dramatic ways.
More on the tumor microenvironment, you mentioned sort of immune response and modulating immune response. Rumor has it you've been working on CAR T cells and their effect. Could you sort of speak to where that is? And
rumor and publications and clinical trial results. So taking a step back, I'm sure we'll get to the synaptic connectivity of these tumors in a minute, but just to say out loud that these cancers are synaptically integrating into the circuits that they invade. And when we discovered that and confirmed it, and I took a deep breath and thought, oh my goodness, boy, are we in trouble. How are we ever going to fight this thing that's kind of actually becoming part of the brain? I took a deep breath and thought, okay, maybe cancer cellular therapy is the way. Maybe if this cancer is really like glitter in jello, it's becoming part of the nervous system. Maybe a targeted cellular therapy could piece by piece take the glitter out of the jello in a safe way that leaves the rest of the brain intact, and was really encouraged and excited by the CAR T cell results in hematological malignancies that were evident at the time. So about 10 years ago, my lab started looking for immunotherapy targets on diffuse midline gliomas, found just a wonderful target, something that is uniformly highly expressed on every malignant cell, and started to collaborate with a really wonderful, scientist, clinician scientist and pioneer in CAR T cell therapy here at Stanford, Crystal Makel. And we found that her CAR T cells targeting this thing we found on the surface of DIPG and other DMGs called GD2. It's a disalyganglioside. It's a glycolipid. That we could cure mice with patient-derived DIPG. So recognizing that it's always more difficult to cure a mouse than a patient, we brought this to clinical trial and have seen in some patients really incredibly hopeful responses, including a boy with a bona fide DIPG, bona fide biopsy-proven DIPG that has had a complete response, complete disappearance of the tumor, evident now for about 4 years. He's thriving. He's doing wonderfully. He just went off to college. I mean, it's just, this, what I'd like to achieve for every single patient, but it hasn't been as durable or as complete for the majority of patients. And so trying to understand that and understand what might be modulating the immune response in the context of the nervous system. Of course, I have been asking questions about how the nervous system itself influences immunotherapy. And these are, ongoing, very active projects that hopefully by the time we have the symposium in 2026, I'll be able to talk about, but really, really actively trying to understand how neurons, neurotransmitters, neuropeptides are potentially influencing the anti-tumor immunity activity of CAR T cells.
And I want to turn that around and talk about the other side of that. Recently, you showed that the immunotherapy can also affect the nervous system. So can you talk a little bit about the role of cancer neuroscience and chemo brain and other side effects?
Yeah, so part of the scope of the field of cancer neuroscience, of course, is to understand not only how the nervous system influences cancer, but how cancers and our therapies for cancer influence the nervous system. Something I've studied for about 25 years, actually since the time of my PhD, is how cancer therapies result in long-term cognitive symptoms. And what consistently, becomes the center of everything we study and every pathophysiological mechanism we identify, reactive microglia in response to the cancer therapy, in response to radiation, in response to traditional chemotherapies, now in response to immunotherapies like CAR T cell therapy, causes dysregulation of other neural cell types and prominently causes a loss of myelin homeostasis and plasticity. And this is a dysregulation. It's not a destruction. It's reversible. And it's reversible with experimental interactions like depleting microglia transiently and allowing them to repopulate in a homeostatic state. But excitingly, in studying this pathophysiology and this cascade of cancer therapy-induced microglial reactivity-induced dysregulation of cell types like oligodendrocytes, we've discovered that chemokine signaling is central to this, and that using a multi-chemokine receptor blocker against the chemokine receptor CCR3, we can normalize microglial cell state and rescue both the cellular and the cognitive phenotype, at least in mice. So I'm very excited about this because I think that it's a mechanism that seems to be common amongst multiple different cancer therapy-induced cognitive impairment, syndromes. We see this after multiple kinds of chemotherapy. We see this after CAR T cell therapy. Interestingly, we see the same pathological cascade after other peripheral immune challenges, including after respiratory COVID and influenza, white matter specific axon tract microglial reactivity, and consequent loss of myelin homeostasis and plasticity. If it proves that CCR3 blockade rescues in these other disease contexts, and that's something we're studying right now, then we have an answer for what is a very widespread problem of long-term cognitive impairment symptoms after these various kinds of immune challenges. But after CAR T cell therapy in particular, we know this works in mice, and so the question is, how best to translate these findings for patients who are suffering from long-term impairments in attention, concentration, memory, multitasking, speed of information processing, et cetera.
Super, super interesting. I'm just thinking about what you said about synaptic integration and sort of the adaptive immune response with CAR T cells. And when I think about, like, in the context of aging, there's synaptic pruning that occurs. You start to lose these connectivities, and that perhaps is in part due to innate cell processes like macrophages. And so I just wonder if, in addition to the CAR T cell approaches, are you thinking about using innate strategies to complement the adaptive?
It's interesting when we study all of our correlative data from the CAR T cell trial, implicates myeloid cells as a problem. It implicates that myeloid cells and their response to the CAR T cell therapy, may in many phases of the immune response be somewhat immune suppressive to CAR T cell efficacy. Possibly contribute to some of the neurological symptoms that we see both in the short term and in the long term. So far, and like myeloid-derived suppressor cells, there's certainly a myeloid-derived suppressor cell phenotype that is evident in patient CSF after CAR T cell therapy, and we reported this, even evident in just even the first few patients. It comes up later in the course after CAR T cell treatment, around the time that we start to lose CAR T cell levels and persistence and decreases, and we see decreases in efficacy for that infusion. So it correlates very strongly with loss of activity. We're working on understanding, how those MDSCs and other kinds of myeloid cells might be suppressing CAR T cell function and how we might target that in ongoing work. But in addition to that, myeloid-related chemokines have correlated with patient, neurological symptoms in the short term. When they have tumor inflammation-associated neurotoxicity symptoms, transient worsening of existing neurological symptoms, et cetera, this is when we see elevation of, myeloid-associated chemokines and cytokines. So in addition to the long-term effects, for which we know microglia are central, which is what, the brain resident myeloid cell, I think that there's likely to be important contributions of other myeloid cell types in preventing effective CAR T cell therapy, and something that, we and others are studying in a very focused way.
Kenzie, you were about to ask a question?
I was. You actually mentioned something about synaptic pruning, and my first response to that as well. We just now start to have the multiphoton in vivo imaging to let us see that a lot of what we thought was bona fide pruning may just be a transient interaction or shielding. So I wanted to ask Dr. Monje about what are some of the techniques you think, especially borrowing from traditional neuroscience, what do you think we can apply to cancer neuroscience to really get at some of these interactions between cell types?
I think that it's clear that microglia are regulating synaptic persistence and, regulating neuronal connectivity and excitatory to inhibitory balance. As we have more and more live imaging, this synaptic engulfment is clearly very important, whether that means that the entire synapse is pruned or whether that engulfment then later results in some other mechanism of synaptic removal. Clearly, microglia are crucial in this process. And how glioma cells or other brain cancer cells influence microglia and possibly alter that very important role in synaptic biology is a really pressing question. And one could imagine that the excitatory to inhibitory balance might be really different in the context of tumor associated microglia or glioma associated microglia than in the healthy brain. So more to come, I think, on that, but it's a really important area for study. Thank you for asking about it.
I wanted to just go back to something you said, just in terms of like model systems, and a lot of work you've been doing in rodent model systems, along with sort of human biopsy samples. And I just wonder, there's been discussion of, and you also mentioned the heterogeneity of response in the context of COVID and flu, and how it's a very complicated situation, right? And the mouse models, to what extent they can sort of recapitulate that sort of heterogeneity you see in human cancer phenotypes is a question. And so, with the new technologies like organoids and such, do you feel, and also the recognition now that immune response that's elicited in murine models and in rodent models is not exactly the same. There's more of a myeloid bias, perhaps, in mouse models. Where do you see the future of sort of these model systems moving? Do you see them as organoids on a chip? Do you see them as modified rodents? I mean, I know the NIH is talking about phasing out of animal models. So just your thoughts on where we see these experimental systems going. Are they going to just really be sort of ex vivo human studies?
So I think that, and I'm trying to remember who originally said this, but it paraphrases to no model is perfect. Many models are very useful. And I have found that, the discoveries we've made about the way that the nervous system interacts with glioma cells in mouse models has really been validated in human tissue and in patient studies. So it has been predictive. I don't think that there is any way to study complex neural circuitry and its influence on a cancer outside of an in vivo system. I do think that there are very useful co-culture and organoid co-culture kinds of experiments that allow you to reduce a system to two components and ask mechanistic questions. But it's hard to study these interactions outside of the complexity of a functioning nervous system. So I think that, taking a multi-perspective, multi-model approach is going to be the most informative, using the wide variety of patient-derived tumor models and testing a particular question using multiple different patient-derived models to understand the generalizability of a finding or the nuanced heterogeneity is really important. I think that using a mouse model with an intact immune system gives you another set of, of answers that are important to consider together. I think that an intact nervous system and understanding complexity in the big picture is really important. And I think that, taking a reduced system and looking at 2 cell types in co-culture or 3 cell types in co-culture can help you really disentangle mechanism. If you look at any of the papers from my lab, we do all of the above. And so, and this is why things take 5 to 7 years to come out, as we look at it every way we can. And whenever possible, validate what we find in patients. And increasingly intraoperative electrophysiology, direct from patient samples have been incredibly informative, but have really validated what we have seen in these mouse models. So it's hard for me to imagine getting rid of mouse models anytime soon. There's just no way to ethically recapitulate that, complexity.
I know that there's been work on heterogeneous organoids, and they sometimes just don't look like they really reflect, the
They're
complexity of.
useful in some ways, but it's not a functioning neural circuit with behavioral outputs.
Right.
It's just not a brain. So,
Right.
I like organoids, I really do, and I also
Yeah,
like
yeah.
complex
We're not
organoids.
picking on
They're
organoids here.
useful.
Many of the people,
They're
but
useful in a lot of ways, and they're experimentally tractable in ways that a living rodent isn't. Sometimes you really need to understand anatomical contributions, every cell type, working neural circuits, effects on behavior. You can't do that ex vivo.
it reminds me of your roadmap concept of cancer neuroscience and how it sort of requires multiple levels
Yeah.
of integrating analysis and not just relying on one sort of perspective, but thinking more broadly. So where are we in that roadmap? As we approach the 2026 Cancer Neuroscience Symposium.
So the roadmap is ever evolving. I think that the scope of the field, from the original roadmap in 2020 to sort of the updated roadmap in 2023 that we sort of collectively and collaboratively wrote as a field, expanded, from a figure, describing the scope of the field that had 3 boxes to a figure that had 6 boxes. But in general, the different ways that nervous system cancer interactions are important, and increasingly recognized, this needs to subsequently be translated to improve therapeutic strategies. And I think that my and many other people in the field have an increasing appreciation that cancer neuroscience is really a hallmark of cancer. It's really one pillar of oncology that needs to be studied and incorporated into treatment strategies to effectively treat some of the most difficult to treat cancers. When we understand that not only are these interactions promoting cancer growth, but protecting the cancer cells from other therapies, it becomes perhaps one required component of a multimodal therapeutic strategy. And so understanding, the granular details of what each tumor requires in order to disrupt these, crucial nervous system cancer interactions and leverage those insights for better therapies is something that I think, we and many others in the field are eagerly working towards.
Fantastic. I think that's probably a very good place to stop. And I just want to say thank you so much, Michelle, for taking the time to speak with us, and we look forward to seeing you at the conference in Texas.
I look forward to it too. Thank you.
Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.